Hello and welcome everyone. My name is Shirsha and today we are going to talk about the different types of page. So this lecture is essentially an extension of our previous lecture where we talked about SDS page and what are its utilities, what is the basic principle, how the gels are made, so on and so forth. In this particular lecture, we are going to cover and talk about the different types of polyacrylamide gel electrophoresis that are commonly used in laboratories. So to begin with, we have native page. Now native basically means in the state in which something actually is without any modification. So what does native page entail? Native polyacrylamide gel electrophoresis, in contrast to SDS page, is run in non-denaturing conditions. So mind you, if you can recall our previous lecture, then you will remember that one of the critical steps in SDS page sample preparation was denaturing the sample. And for denaturing the sample, we use a lot of different techniques like heating, treatment with SDS, which is actually an anionic detergent, treatment with reducing agents. But in a native page, in contrast to that, there is no denaturation of the sample being run. Complexes therefore remain, for the most part, associated and folded as they would be in the cell. So in native page, however, complexes may not separate as cleanly or predictably as in SDS page. Why? Because since they are not denatured individual proteins, they face more resistance while migrating through the polyacrylamide gel. So in SDS page, the entire aim was to resolve proteins solely on the basis of their molecular weight. We did not want their tertiary structure or their association or interaction with other proteins to play any role in this resolution of the proteins, and therefore we denatured the proteins and coated them with a uniform negative charge. But in native page, there is no denaturation of the protein. Why? So this will allow us to detect any complexes between two proteins if they are formed. Now, due to the specific properties of the prepared gel as well as the electrophoresis buffer solution, which typically has a pH of around 10 and contains tris HCl and sodium azide NaN3, most proteins of a biological system are charged negatively and will therefore migrate from the cathode to anode. So this part remains the same as in an SDS page, wherein in SDS page, the proteins were also migrating towards the anode because of the negative charge imparted by the SDS molecules coating the protein. And over here, because of the pH conditions of the buffer that is used in a native page, the proteins also maintain a negative overall charge, which ensures that they migrate from the cathode to the anode after voltage or electric field application. So it is useful for studying protein complexes. So if you look at this particular image, you would see what a native page looks like. So let's compare between the native page and the SDS page. So essentially the same samples are run on the SDS page and native page. Like you can see SSOB here, SSOB. So in native page, you see the band is coming under at SSOP at a much higher molecular weight of 80, whereas in the SDS page following denaturation, the band you're getting is at around 20 kilodalton. So what does this show? That probably this band that we're obtaining over here is actually a tetramer, and over here we have the monomer which we obtain upon denaturation, because the molecular weight is roughly 4 times of what we observe in the SDS page. Similarly, if you look at this particular lane, say for example BL21, now, I will not go into the details of what each of these lanes mean. It has been taken from a research paper which has been cited here. But this is just to convey the message as to how a protein resolves or how a complex of proteins resolves differentially on a native page compared to an SDS page. So, in this particular lane, if you compare BL21 on native page and SDS page, you would see that the band that you're obtaining is around 480 kilodalton in molecular weight if you compare it to the molecular weight marker, whereas in the SDS page the band that you're getting is roughly around 20 kilodalton. So in a native page, the tertiary or the quaternary structure of a protein plays an important role in determining how the protein will migrate through the gel. So coming to the different types of native page, within native page also there exists some variation. So we have clear native page or CN page. So what is that? CN page uses no dye and hence the name clear, and this therefore makes it difficult to examine the separation of proteins. After the complexes are separated, the gel is cut and the protein complexes can be extracted from each portion separately. Because you're not using any dye to stain the protein, so therefore the protein remains in its more or less native state, and therefore you can extract the protein complex from the gel and you can use it for further analysis. Because you're not denaturing it, so the complex is intact and the interactions that are present in the complex are intact. Because you're not staining it, it's not going unnecessary undue modifications, and therefore you can do further studies with these protein complexes post extraction. Now, each extract may then be, for instance, sequenced, or it can be examined by mass spectrometry. So, for example, say you have a sample and you want to, it's a mixture of, say, multiple proteins, and you want to identify what are the proteins present in that particular sample or what proteins form a complex. So, in that case, you would run a clear native page. So, this will separate out your complexes based on the molecular weight, and now you can isolate each individual complex from the gel by extraction and then analyze and identify what protein that is, either by sequencing or by mass spectrometry. Now, CN page is therefore useful for examining protein-protein interactions. Just like any other page, CN page is also useful for studying protein-protein interactions. The next type we have is blue native page or BN page. So BN page is more commonly used compared to CN page, and it's simply a modification of CN page wherein the gel is stained with Kumasi blue, which allows the separations to be examined with the naked eye. Because you're now staining the protein, you can now visualize the protein with your naked eyes. So if you take a look at this image, you would see a blue native page of the same samples compared to a clear native page of the same samples. So you see, for clear native page, if your sample, for instance, comprises of proteins that by themselves fluoresce a certain color or which by themselves have a certain color, in that case, you can easily visualize them on the gel without the need for any staining. Because obviously, if you don't know where your protein is on the gel, then how will you go for extraction? You can't randomly cut out a portion of the gel and extract a protein. You need to know where your protein is residing in the gel. In this case, you would see that thylakoid protein complexes are on the gel, and because, as you know, thylakoid contains the green pigment chlorophyll, so therefore you can easily visualize them without the need of any staining. And this is simply the same gel which has been stained, and this will be your blue native page. There is another kind of page which is known as QPNC page. Now, QPNC page or quantitative preparative native continuous polyacrylamide gel electrophoresis is a high resolution technique which is used to separate proteins by their isoelectric point. Now, what is the isoelectric point of a protein? The isoelectric point of a protein is that particular pH at which the protein has a net charge of 0. And we will be discussing this in detail in the coming up slides. So QPNC page procedure is accomplished typically in a special electrophoresis chamber. Although the pH value of the electrophoresis buffer does not correspond to physiological pH value, which is typically around 7, the proteins are eluted continuously by a physiological buffer solution which has a pH 8 and isolated in different fractions. So the buffer pH is different, but this entire setup of QPNC page is actually a bit more complicated, and it is less commonly used in a day-to-day basis in laboratories. It is used only for very specific studies. So the protein is eluted at a pH which is close about the physiological pH, which ensures that the functionality of the protein is maintained. Now, the separation system, including the electrophoresis chamber and a fraction collector, are all cooled in a refrigerated chamber. Why? To ensure that the protein can retain its functionality for further subsequent studies. The pore sizes of the polyacrylamide gel are very large, and therefore the sieving effects become minimal during the electrophoretic separation. Obviously, if your pore sizes are small, then huge complexes will face more resistance and more difficulty when migrating through the gel. So large pore sizes are typically preferred. QPNC page is used to isolate active or native metalloproteins in biological samples, and to resolve properly and improperly folded metal cofactor containing proteins in complex protein mixtures. So this is actually a very restricted and common application of QPNC page, whereby you want to study metalloproteins or proteins which contain any metal ion as a cofactor. So over there QPNC actually becomes very useful. Other than that, the applications are pretty limited. The bioactive structures, that is the native or the three-dimensional conformation of the isolated protein molecules, do not undergo any conformational changes when using QPNC pH, and that is the entire point. And that is why you maintain a physiological pH in the elution buffer and a cold temperature of 4 degrees C to ensure that the activity of your protein and its native structure is retained. And consequently, quantitative amounts of highly purified metalloproteins and protein isomers are isolated in the different fractions. So hence it's a quantitative and preparative page. Why? Because you can quantify the amount of protein that you're isolating from this method, and it's a preparative scale procedure. That is, you can use this to produce large amounts of protein. It's not an analytical procedure whereby you just want to analyze something. It's a preparative procedure whereby you can produce large quantities of protein by this particular method. So this is typically what a QPNC page setup looks like. You can see that there is like an outer chamber and an inner chamber, and you see this narrow part in the center. This is basically where your gel lies, your polyacrylamide gel lies, through which your protein will be passing. And then there are like motorized pumps fixed with it, which helps in maintaining a particular pressure with which your protein sample will pass to the gel, and so on and so forth. And like, let us not get into the details of it because then it would become an entire lecture of itself. So the next kind of polyacrylamide gel electrophoresis is zymography. Now, what is zymography? It is an electrophoretic technique based on SDS page that includes a substrate copolymerized along with the gel for the detection of enzyme activity. So over here, not only do you want to separate your proteins on the basis of their size, but you also want to detect their functionality in the same gel. So it involves the electrophoretic separation of proteins under denaturing but non-reducing conditions through a polyacrylamide gel copolymerized with a protease substrate. So typically the kind of enzymatic activity that you want to detect in zymography is protease activity, and so therefore the proteins or the enzymes that you're looking for are proteases, which basically degrade other proteins. That's what a protease is. So you run this gel under a denaturing condition. So this will ensure that your complexes are dissociated from each other, but it will ensure that the protein is coated with an overall uniform negative charge, and migration is solely on the basis of its size, and its charge by itself plays no effect on migration, but non-reducing conditions. This is because the final fold of the protein is very critical for its functional activity. Now, if you reduce these disulfide bonds, then you cannot reverse back the process as easily as you can reverse back denaturation. So therefore, your gel is run under denaturing but non-reducing conditions. Note, reduction and heating would damage the enzymes and acids, so these should be avoided at every cost. So gelatin and casein are commonly used as protease substrates. The resolved proteins are renatured by exchange of SDS with a non-ionic detergent, for instance Triton X100. So therefore this SDS denaturation is reversible and is acceptable in zymography, but you would have to be very critical and you have to ensure that you avoid adding any reducing agent, or you have to avoid heating your sample. The gel will then be incubated in an appropriate buffer for the proteinases under study and visualized by staining with Kumasi blue. So if you take a look here at the gel, you can see that the entire gel is getting stained blue because of the presence of the protease substrate, which in this case is gelatin. So it is taking up the stain and it's getting stained blue, and only in the regions where you have enzymatic activity do you obtain clear bands. So this particular figure represents the MMPs or matrix metalloproteinases that have been separated by running a polyacrylamide gel electrophoresis. And this is a standard, which is basically a molecular weight marker, and the molecular weight marker itself comprises of proteins of different molecular weights, known different molecular weights, which also have the enzymatic activity. Because if the marker itself does not have the enzymatic activity, you cannot visualize a marker on the gel. And if you cannot visualize a marker, then you cannot determine what is the weight of the proteins that are being separated out on the gel. So protein-like activities are detected as clear bands against a blue background of undegraded gelatin. So next we're going to discuss about isoelectric focusing. But before that, I would like to talk about two important concepts. A zwitterion. What is a zwitterion? So as we know, amino acids have an amino group, a carboxyl group, and an R side chain. So depending on the pH, the carboxyl group can be either protonated or deprotonated, and the amino group can also be protonated or deprotonated. So this form is the non-ionized form of an amino acid. Now this form of the amino acid is an ionized form, whereby the amino group is protonated and has an extra proton and therefore an associated positive charge, and the carboxyl group is deprotonated, that is, it has lost this proton. And in this particular state, the net charge of the amino acid is 0, and this ionic state is called the zwitterionic state. Now, next is isoelectric point. What is the isoelectric point of a protein? So, as the pH of a solution increases, it has less and less amount of free protons available. So say we start at a lower pH. In lower pH, you have excess of protons available in the solution. So therefore, both the amino group and the carboxyl group are protonated. Now, as the pH increases, after a certain point, which let us say call it the pK1, the carboxyl group loses its proton, and therefore takes on a negative charge, and the amino group is still protonated. Now this state is what we would call the zwitterionic state. Now when you increase the pH even further and there is a further lack or dearth of protons in the solution, the amino group also loses its proton, and now the amino acid is in a negative, has an overall net negative charge. So in the beginning, the amino acid has an overall net positive charge. Next, it is in the zwitterionic form where it has a net charge of 0, and then it is at the third form where it has a net negative charge. So if one is to plot this net charge versus the pH, then this is something that it would look like. So see, this is where it was a cation, then a zwitterion, and then an anion. So at pK1, at pH equivalent to pK1, you see there is this decrease in the net charge, and at pH equivalent to pK2, there is this second change. There is a loss of the second proton and change in the net charge. And this particular point where a particular amino acid or a chain of amino acids, that is a protein, has a net charge of 0 is called its isoelectric point, or PI. Now with this concept, we are going to discuss about isoelectric focusing. So isoelectric focusing, also known as electrofocusing, is a form of electrophoresis for determining the isoelectric pH of proteins and separating different proteins by differences in their isoelectric point. So in isoelectric focusing, proteins are resolved not on the basis of their molecular weight or mass, but rather on the basis of their isoelectric point. So proteins are first introduced into a gel composed of a polyacrylamide or agarose where a pH gradient has been established. So first step of isoelectric focusing is to generate a gradient of pH in a gel. Now, gels with large pores are usually used because you want to ensure that proteins are separated solely on the basis of differences in their isoelectric point, and that their size does not have any effect on their separation. So in order to eliminate any sieving effects, gels with large pores are usually used so that different proteins of different sizes will migrate at a uniform rate, and any separation that you observe will be solely because of differences in their isoelectric point, or PI. Now, pH gradient is established by using synthetically charged ampholytes, which are variously charged aliphatic aminocarboxylic acids of varying molecular weights. So that's the first step. You use these synthetically charged ampholytes to generate a pH gradient. Now, when a potential is applied to the electrodes, the ampholytes will migrate to positions in the column where they're equally attracted by both the anode and the cathode. That is, they are going to arrange themselves in order of the isoelectric pH. So, if you take a look at this schematic, this will help clear things up. So first you have, say, a mixture of ampholytes, right, which are nothing but these variously sized aliphatic aminocarboxylic acids, and when current is applied, they will migrate towards either electrode, towards either the positively charged electrode, which is your anode, or towards the negatively charged electrode or the cathode. They will migrate till they reach a point where they're equally attracted by both the electrodes. That is, they will arrange themselves first according to their isoelectric pH, and this will therefore help generate a gradient of pH in the gel. So a gradient is established in which the pH increases as one progresses from the anode towards the cathode. So when you move from the anode to the cathode, there is an increase in the pH. This gradient is established prior to adding the proteins of interest to the gel. So this is the first and foremost step. So once a pH gradient has been established on the gel, the next step is to load the protein samples on the gel. So if you go back a couple of slides and you remember the zwitterionic slide in which we talked about the zwitterionic stage of an amino acid or protein, then you would remember that as the pH was increasing, an amino acid or a protein goes from a cationic stage to a zwitterionic stage to an anionic stage. So when the protein is in a region where the pH is below its isoelectric point, it would be positively charged because it's in its cationic stage. There is an excess of protons available in the solution, and it will migrate towards the cathode. And when the protein is in a region where the pH is above its isoelectric point, it will be negatively charged and it will migrate towards the anode. This might seem a bit confusing, so if you want, you can pause at this particular moment and think about it, and that will help clarify it further. So the proteins migrating away from the anode become less and less positive. Why? Because again, if you go back to the previous slide where we talked about the ampholyte gradient or ampholyte mediated pH gradient that was established in the gel, you would see that as one was moving away from the anode towards the cathode, there is an increase in the pH. So as the protein migrates towards the cathode, it becomes less and less positive. And as it migrates towards the anode, similarly, it becomes less and less negative till it reaches a point where its pH is equal to, where the pH of the gel is equal to its isoelectric point, and the net charge of the protein becomes zero. Now, once the net charge becomes zero, it is neither attracted towards the cathode nor attracted towards the anode. So electrophoretic migration stops, and this allows proteins that were loaded onto the gel to become distributed throughout the gradient as a series of narrow zones in order of their isoelectric points. Now, once migration of proteins is complete, the separate proteins may be collected as a series of fractions, or they can be used for further studies, as we will discuss later. Isoelectric focusing can resolve proteins that differ in pi value by as less as 0.01. So as you can see, the resolution or the resolving power of this particular method is pretty high. So if we take a look at this schematic, which sums up what we have studied or discussed so far, once you have cast your gel, first you apply an ampholyte solution, and once you have loaded the ampholyte solution, you apply an electric field to your gel. Application of the electric field will help in the establishment of a pH gradient. So a pH gradient is established from, say, pH 9 to pH 3 in this particular schematic, from the negatively charged electrode towards the positively charged electrode. Now, once you've established the pH gradient using ampholytes in your gel, you then stop applying current, you load your protein sample onto the gel, and then you reapply the electric field. So this will now allow your protein sample, whatever proteins are present in your sample, to be separated out on the basis of their isoelectric points. And then you can simply elute the proteins from the different gel fractions. You can extract it from the different gel fractions, or you can just simply stain it to visualize how the proteins have separated out. So next we move on to 2D gel electrophoresis or two-dimensional gel electrophoresis. So this is an extension of isoelectric focusing, I would say. So 2D gel electrophoresis methods have been developed to improve the resolving power of a one-dimensional SDS page procedure. So a one-dimensional procedure is one where you separate out proteins on the basis of only one criteria. Say for instance in an SDS page on the basis of their molecular weight. In 2D gel electrophoresis, you use two dimensions to resolve your proteins. So for example, two dimensions could be pi or their isoelectric point and their mass. So in the first step, the mixture of proteins is electrophoresed through a narrow tube gel containing ampholytes that set up a pH gradient. So essentially the first step is isoelectric focusing, where you separate out the proteins on the basis of their isoelectric point. Now proteins will electrophorese towards the anode till they reach the isoelectric point. In the second step, you take this gel from the tube. So and then you, for instance, if you have say this vertical tube in which the gel is present, you take out the gel from the vertical tube and you place it horizontally on the top of an SDS page. So now these proteins which have already resolved on the basis of the isoelectric point will now undergo resolution or will be separated on the basis of their molecular weight. So you place it lengthwise across the top of an SDS polyacrylamide slab. Now the proteins which have been partially resolved by isoelectric focusing are further resolved according to their sizes by SDS page. And once you have run the gel, you can visualize it either by Kumasi blue staining or silver staining. So proteins that have been separated can be cut out from the gel and then further analyzed by mass spectrometry, which will help identify the protein. So you see, this is where you have the first dimension, where you separate out the proteins on the basis of their pi. Then you take this vertical gel and you place it horizontally on top of a polyacrylamide gel slab, and then you run the gel again. And now you see that these proteins which have already separated will separate further on the basis of the molecular weight. And this is typically what a 2D gel looks like upon staining. So you don't really get very discrete or significant bands like you were getting. I wouldn't say significant, let me rephrase. You wouldn't get distinct bands as you were getting in case of SDS page. But over here you get more of like spots and some bands, but mostly you have these spots that are spread out. That is what typically a 2D gel page looks like. So one application could be, for instance, you have in your protein solution or in your sample, you have two proteins which have the same molecular weight. So you cannot separate them out by using or by doing simply an SDS page. But if these two proteins have different isoelectric points, then you can first separate them out on the basis of their pi values. And then when you run an SDS page, you will separate them out from one another. For instance, say if 2 points lie on the same line, right? Say for example, here this point and here this point, they are lying along the same line. So they have the same molecular weight, but because they have different pi values, they were in different parts of the gel, of the first gel, which allowed them to be separated following running the second gel. Had you run simply an SDS page, then this and this protein would come at the same point. They would be merged and you couldn't be able to distinguish between the two. So I know these concepts seem a bit confusing at first, but I would suggest that you go through them a couple of times. And in case you have any questions, please feel to leave a comment, and I would definitely get back to you. If you like this presentation, do like, share, and subscribe, and keep following for more such presentations. Thank you, and I'll catch you next time.